Hello, this lecture is going to be tissue and wound repair. So it's important to understand that there's two types of tissue and wound repair. First is going to be regeneration, which is the replacement of the destroyed tissue by the same type of cell. So this is if you have a small cut and then you just get new skin kind of on top of it. And then the second type of wound repair is called fibrosis. This is where you're going to have scar tissue forming because there was a deep cut. So we're going to talk about the different steps of tissue and wound repair. The first step is called proliferation. This means that the capillaries are going to become very permeable. They're basically going to vasodilate, which means get wider in diameter. And when it says more permeable or very permeable, it's going to allow different substances to enter and exit a little bit easier, like white blood cells, for example. You're also going to have proteins that are going to get into the area and construct a clot so you don't have any blood loss. And this helps the wound dry quickly and forms a scab. Then you have granulation tissue forming. This is delicate pink tissue composed of new capillaries and lots of white blood cells, along with collagen fibers if you do form scar tissue. This is if you've ever peeled off a scab maybe a little bit too early and the tissue underneath it is really pink and kind of liquidy. The third step is surface epithelium regeneration. So once we've created granulation tissue and we start the healing process, then we're going to have the new tissue forming on top. If you do go through fibrosis and you have scar tissue, then sometimes the scar can be a white or a dark color, that all depends on the pigment of your skin. So now we're going to talk about the developmental aspect of cells and tissues. How do cells and tissues actually form when you are in the womb? So pretty much you start out as a little ball of cells, and as you start to go through mitosis and create more cells, they begin to specialize, meaning that they kind of choose what cell type they're going to be, because a cell is just a cell until we turn on certain genes that then make it a muscle cell or a liver cell or an epithelial cell. And by birth, most of your organs are really well formed and very functioning, and we keep going through cell division and mitosis when we need to make new cells. And pretty much all of your body cells undergo mitosis till you die, except for two types. Neurons, which are in the nervous system, you actually don't have, you don't divide at all. And this is really important because if you think about it, someone with a spinal cord injury or a brain injury sometimes don't recover fully, and that's because they don't go through mitosis. Neurons don't make new neurons. The neurons you are born with are the neurons that you die with. And then after puberty, only certain cells divide. For example, your cardiac cells stop dividing after puberty. So like we just said, after puberty, cardiac tissue does not divide. It becomes ameiotic, which means it loses the ability to divide. And neurons are always ameiotic, meaning that they don't divide at all. Some abnormal cells and tissues can be a neoplasm, which is basically a tumor. If it is a malignant tumor, it's going to be a cancerous tumor. And if it's a benign tumor, it's going to be non-cancerous. Then we have hyperplasia. This is when organs enlarge due to maybe a hormone or an irritant. For example, when you get pregnant, your breasts enlarge so it's easier to breastfeed. And then atrophy is the decrease in the size of an organ. Happens a lot after surgery. For example, you have a lot of muscle atrophy when you get surgery on a certain area. And this is just a picture kind of showing, if you look on the left, that calf muscle is much larger than the calf muscle on the right because this person just had surgery on that leg. So that's atrophy. All right, thanks for watching. Please rewatch if you need to fill out the rest of your note guide, and you are officially done with Chapter 3.